put all of the learning objectives from those chapters that we covered up here. So the learning objectives are a really good starting point for asking yourself if you even have some sort of concept of what sort of information you should be trying to think of. Right? So for example, explain how mitosis results in genetically identical cells using terminology like chromosome, chromatid, and chromatin. Anybody tell me what chromatin is? Okay, so, right, learning objective number one, right, when you look at that, what you should say to yourself is, well, I don't know what these terms are, so I should probably go do some reading and figure out what these terms mean. Right? That's why I say the learning objectives are a really great place to start as you study for the exam, right? So, first things first, right, just being familiar with what's being asked for in the first place, right? So a chromosome, right, a chromosome, I'm going to ask somebody to come up to the board, but that would just take forever. I think we all do that. Plus, I'll have to wait for somebody to be brave enough to do it. A chromosome, is that a chromosome? It's not. Why not?
It's in pairs. There's like supposed to be two.
Yeah, always? Is it some of the time? Only specific times?
Sometimes.
This is a chromosome, right? This is what a chromosome usually looks like, right? Chromosomes almost always look like this, right? If I draw it like this, what is it now? Not just a chromosome. Well, the two components of that chromosome are sister chromatids, but what do we call the chromosome itself? There's even a process related to the production of that second arm. You might call that process replication. So this would be called a replicated chromosome, right? This is a replicated chromosome. It only replicates in preparation for what process? Mitosis, right? Cell division, right? That's the only time we need to replicate that, right? So chromosomes don't usually look like this, right? They only look like this if they've been replicated in preparation for cell division, right? A normal cell, just doing its day-to-day thing, is just going to have a chromosome like that. Would there just be that one chromosome, or would it have a partner? Yeah, right, because we have two copies of every chromosome, right? What do we call those two chromosomes? We just call that a set of chromosomes. Right? So there we have a set of chromosomes, right? What other word could we add to that? A set of blank chromosomes. Are they the same as each other? They are, right? So we call them, this is a set of homologous chromosomes. Right? Two homologous chromosomes mean they have all the same genes on them. Right? Will those genes necessarily be identical to each other? Not necessarily. Right? They could be identical to each other, right? Or you could have a couple of variants. Right? And if we have variant number 1 and variant number 2, it's the same gene, right? But those genes aren't identical to each other because one of them might have a mutation that makes it slightly different, right? It's a different version of that gene. In which case, right, we would call these, what do we call two versions of the same gene? Alleles, yes. So alleles are two versions of the same gene, right? So homologous chromosomes are going to have all the same genes, but they may not have the same versions of those genes. Right? They might have different alleles. The location of those alleles is called what? Even sounds look like location. That is a locus. Right? A locus is the spot where we find a particular gene, right? And then look for the variants of that gene, which are called alleles. We already covered chromatids, right? So when these chromosomes replicate in preparation for cell division, right? So now we have a set of homologous replicated chromosomes, right? And we call each individual one a sister chromatid. And those chromatids will be pulled apart at some point, right, into separate cells. And once they get pulled apart into separate cells, right, then they'll go back to our original single chromosomes. That leaves us with chromatin. Right? Chromatin is just DNA, just protein, or a combination of DNA and protein. You have a 33% chance of getting this right. Did you say a combination? Yeah, it's a combination, right? It's DNA wrapped around proteins called histones, right? We call that chromatin. Right? Remember, DNA, right, doesn't just kind of sit loose inside the cell nucleus, right? It's very organized, right? The DNA itself is a double helix, right? That double helix is wrapped around histones. Those histones then are stacked on top of each other, and so we have this coiled coil of DNA. And then all of that, right, finally gets all those histones packaged together until we have that structure of the chromosome that we see there. Right? But if we unravel that, right, we'll see that that DNA is wrapped around those histones, and that's what we call chromatin. All right. What are the events that happen in interphase? In interphase? That's right. Be sure of yourself. Yes, an interphase, right? Interphase has 3 subphases. What are they? We're talking about interphase. Let's back up a step. The life cycle of a cell, the entire life cycle of a cell has how many phases? How many primary phases? Did I hear somebody saying timidly 2? Right? The 2 primary phases are interphase and the mitotic phase. What are the 3 subphases or stages of interphase? There we go. G1, S, and G2. Very good. And what happens during G1? Yeah, just a cell growth stage, right? We want to make sure that we have enough stuff to split. What happens during S? There's your replication, right? So that's the synthesis, right? S stands for synthesis of DNA, right? Because we are going to replicate all of our chromosomes. And then what happens in G2? More growth. Let's see here. And then the mitotic phase is split up into what stages? Yeah, the ones we're familiar with, right? Prophase, prometaphase, metaphase, anaphase, telophase, and then finally it ends with cytokinesis. Let's see here. 3 checkpoints. Anybody remember where those 3 checkpoints are? Yes, G1 has a checkpoint, right? If we take a cell that's in G1 and we combine it with a cell that is in S, what will happen? They will be combined, yes. And we have one cell that's in G1 still. We have one cell that's moved on to S phase. It's tested checkpoint. We fuse those cells together. What does the first cell do that was in G1? Does it stay in G1? Does it move on to S? Yeah, it goes to S, right? Because those checkpoints are due to cytoplasmic factors, right? Things that are inside the cell. So if we have a cell that's in S, that means that it has all the cytoplasmic factors to have passed that checkpoint and be in S. If you fuse that with a cell that's in G1, it'll share those factors with the cell that's in G1, and that cell will go ahead and progress to S as well. Second checkpoint, right, is in G2. Right? In other words, the cell is not going to waste time, right, moving its centrioles to the 2 poles, producing a mitotic spindle, attaching to the kinetochores, right, moving the chromosomes around. Like, that's a huge investment of energy. And if the cell's not ready, right, it doesn't have all the stuff that it needs to do mitosis successfully, then it will not progress out of G2. If it does have all those things, right, then it'll go ahead and it'll move into mitosis. Let's see here. What's this down here? Can anybody explain this diagram to me, kind of what we're seeing there? So one of those checkpoints, yeah, go ahead, Lou. It is. You've identified it. That's a good step in the right direction, right? And then to explain it, right, one of those cytoplasmic factors, right, is the maturation promoting factor or the mitosis promoting factor. You can call it either one of those things, right? That increases, right, in activity as we get more and more and more cyclins, right? So these little proteins being produced are called cyclins, right? When you get to a critical number of cyclins, right, they start combining with the CDK. That creates your mitosis or maturation promoting factor. Right? That is a kinase. Right? And that kinase then will phosphorylate a variety of things that need to be phosphorylated, right? Enzymes that need to be active in order to move centrioles, right? To produce the mitotic spindle, to attach the chromosomes, et cetera, et cetera. Right? We know right, from earlier in the semester right, that activity of an enzyme is typically due to it being phosphorylated. Right? And so CDK right, is one of those kinases that's activated by increasing amounts of cyclin. Right? It's a cyclin-dependent kinase, that's why it's called CDK. Right? When cyclins have risen to the appropriate level, right, then the CDK will combine with that, it'll become active, and it'll start phosphorylating all the things that need to be phosphorylated so that mitosis can happen. Right? If the cyclins aren't at a sufficient level, right, if the cell has not produced enough cyclins yet, then you won't have right, CDK that's active, it won't be able to phosphorylate things, and so we won't be able to activate all those enzymes that are necessary right, to move the centrioles, to produce the mitotic spindle, and to move the chromosomes around. Right? This is one of the cytoplasmic factors right, that is necessary for a cell to pass that checkpoint. Right? Do I even have enough cyclins to activate the kinase that's going to activate all these enzymes that I need? Speaking of replication, right, if I were to give you, right, what do we call this, first of all, right? It's written up there, but if you were to look at that, right, what would you call that? What? The whole thing. Well, that's the process you see there, but what's the structure? It's a replication fork. Right? If you look at that, right, you should think replication fork, right, which is actually right here where the DNA has actually been split open. Right? That's the fork. Right there, and so when you look at this, you say, oh, this must be replication because I see the replication fork. Replication fork has lots of things. Right? It has this handy-dandy little protein over here, right, called, anybody know what that protein's called? It's the only one that's not labeled, actually. What is the protein that opens up or unwinds the DNA and allows replication to happen in the first place? Did you say sheep? Starts with an H. I even told you all a funny joke, an inappropriate joke about the name of this enzyme. It's called helicase, right? DNA helicase. I wish I was DNA helicase so I could unzip your genes. That's what it's doing, right? It's unzipping the DNA there, right, to allow right, all these things to attach. We have, right, starting, let's start with the leading strands, right? Can the DNA polymerase just attach on its own, or does it require something to be there first? That's right, it does require something to be there first, right? There's got to be a primase. Right? So primase puts a primer there. That allows DNA polymerase 3 to bind, right, and start replicating. Right? And it just goes along down the leading strands. Right? It's much more complicated on the lagging strands, right? Because the lagging strand, right, the primase sets the primer. DNA polymerase will read that direction for a little while, right? And then it'll run into the one that it produced before, and it'll have to jump back and do it again, right? So it had to work its way this way, and it had to jump over to here and work its way that way, and it had to jump back and work its way this way, and it has to jump back to where this primase is setting one, and it'll work its way up to that, and then so on and so forth, right? That's why it's called the lagging strand. It's always a little bit behind because it has to move in the opposite direction. Because you can only move from 5' to 3', We're always adding nucleotides onto the 3', and since the two DNA strands go in opposite directions, right, one of them will just be the leading strand, it will just replicate all the way down that strand, but the other one, since it goes in the other direction, right, it can only replicate that way, and it has to jump back and replicate that way so it can catch up with the leading strand. So, right, we have our primase, right, DNA polymerase joins, right, it replicates up until it runs into the previous primer. Right, then you have DNA polymerase 1. It's going to take out that primer and put in the right nucleotide, and then DNA ligase right, fixes that little split right there. So it's always a little bit behind, right? This DNA polymerase is just moving from its primer all the way down in that direction, right? But this one, right, it started over here and went this way, and that way, and so on. Those are called Okazaki fragments. The only reason we can add to the 3' end, it is a dehydration reaction. Right, remember 3' is and 5' just refer to the carbon number of the ribose, right, that makes up that nucleotide. Right, and so we have carbon 1, 2, 3, 4, 5. Carbon 3 has the hydroxyl on it. Carbon 5 has the 3 phosphate groups on it. Right, and so when we combine those together, right, the adenine is going to be towards the middle. Right, it's going to pair up with a thymine on the other side, right? Adenine and thymine. And above that, you might have cytosine and guanine, maybe guanine and cytosine. And to join these, you have the OH at the 3' ends, the phosphates at the 5' ends, right? And we can add on where that OH is because it's a dehydration reaction. Right? So we get one hydrogen coming off there, one OH coming off there. That's the water molecule in that dehydration reaction. Then we have that, this carbon is bonded to that hydroxyl there. It can't work the other way because you wouldn't have a hydroxyl for that dehydration reaction. Let's see here. Who can name an example of asexual reproduction? Cell splitting by mitosis. That would be asexual reproduction, right? Because you just end up with a clone of that same cell. Right? It didn't involve another partner. There was no genetic recombination. Right? A cell just underwent mitosis. Right? You get an identical daughter cell that results from mitosis. Right? So that is reproduction, but it's asexual. Right? You can name an example of sexual reproduction. Yeah, gametes. Right? So an egg. Or a zygote being created by fertilization through an ovum and sperm, right? The ovum and the sperm are haploid or diploid? What does haploid even mean? Almost like y'all haven't studied this at all. 0 time spent studying for biology, I see. A human has 46 chromosomes in every cell except for the gametes. Right? 23 sets of chromosomes. Right? That makes it diploid because it has 2 sets of, has 2 of every chromosome. Right? The ovum and the sperm only have 1 of every chromosome. Right? They only have 23 chromosomes. That's haploid. Right? Haploid is half the number of chromosomes. Diploid is the full number. We have 1 of every chromosome versus 2 of every chromosome. Let's see here. Who can name an organism that has a different type of sexual life cycle than humans do? Fish produce eggs, which are fertilized by sperm. Same deal. I think of something that doesn't reproduce by producing eggs and sperm. Bacteria do not reproduce by sexual reproduction at all.
It's different.
I said sexual reproduction. That's another form of asexual reproduction. Since you brought it up, what do we call that? It's not called mitosis. It's called binary fission. You can name an organism that does a different type of sexual reproduction than most animals. Do plants sexually reproduce? In what's different about a plant versus a human? It's called alternation of generations, right? They produce a spermatophyte and they have a gametophyte, right? Two different organisms. Sometimes that second organism is attached to the original, sometimes it's not, right? But a plant, when it wants to reproduce, it produces a flower. Right? When a human wants to reproduce, it doesn't have to produce a flower. Right? It has its gametes already on board, right? But a plant, right, has to produce a flower. That's a separate structure, right? That's the gametophyte, right? If that gets fertilized, right, it grows, it produces a seed. That seed leaves the plant, lands somewhere, it grows into a new tree, and that's the spermatophyte. Let's see here. The word zygote. I used it a minute ago, but does anybody know what the word zygote means? The zygote is the fertilized ovum, right? So you have the ovum, haploid, you have the sperm, haploid. As soon as the sperm fertilizes the egg, we call that, or the ovum, right? We call it an egg or a zygote. Let's see here. How is meiosis different than mitosis? Just name anything, literally anything. Yeah, what's that called? You're right. Crossing. Just crossing over. Yeah, crossing over is a feature of meiosis not seen in mitosis. What does that mean, crossing over? Okay, say it loud enough for me to hear though. One what? The whole chromosome? Yeah, right. They swap, not even necessarily full alleles. Well, full alleles, yes, but not necessarily just one allele. It might be a couple of alleles. Right? What they swap, right, is just parts of their arm. Right? So analogous parts of their arms, right, which might contain 2 or 3 genes, might only contain 1 gene. Same place as what? Oh yeah, they have to, right? Because they have to trade the same gene, right? Yeah. But the amount of genetic material that gets traded is variable. So it might just be the 1 gene that gets traded. It might be several genes.
You can call that locations components.
Yep. Maybe now would be a good time to jump into this worksheet.